Should I start or we can wait for?
Okay, Assalamu alaikum and very good afternoon, everyone. Thanks for joining this session, and special thanks to our speaker, Dr. Mohammed Atif Yaqub from the Institute of Photonics, Barcelona, Spain, for joining this session and sharing this talk. Okay, so before we can start the talk, let me read the brief biography of the speaker. So Dr. Atif is a researcher at the Medical Optics Group at the Institute of Photonic Sciences, Barcelona Institute of Science and Technology, Spain. He received his BSc and BSc in Electrical Engineering from Center of Advanced Studies, CASE, in Pakistan in 2008. And he did his master's from Universiti Teknikal Malaysia in 2013. Then he obtained his PhD in mechanical engineering from the Busan National University, South Korea in 2021. And he has over 8 years of academic research and industrial experience, both at universities and in private institutions. So currently he's doing research on different topics related to the brain connectivity near field. So today he's going to share his experience on one of his topic. So without any further ado, let us invite our speaker to start the talk today.
Thank you, Dr. No. Okay, so I'm Atif and I will be presenting the effects of brain stimulation in the brain connectivity. So we will be going through the exploring changes in brain connectivity with near-infrared spectroscopy devices. So I will go through it very slowly, and in case of any questions, please, you can stop me anywhere you want. So these are the contents. I will first introduce the terminologies that I will be using, the technology that I will be using, and functional connectivity, brain stimulation, and then I will be talking about the research that we found the effects of stimulation on functional connectivity and how can we control the stimulation duration using functional connectivity. And then if as the topic also includes new nurse devices, so I will take a chance, if we have time, I will show you the devices that I have been developing. So what is functional near-infrared spectroscopy? So it is an optical brain monitoring technique which uses near-infrared spectroscopy for the purpose of functional neuroimaging. So here in this picture, you see these devices can be bulkier, but like there are small optodes that can be light source or detector on the head of the person, and then we can monitor the activation that are being done in his brain. So how it works, we can go further. So while I'm talking to you, or you're talking to your friends over a cup of coffee or anything, so at the back end of in the brain, neurons are working in the brain behind every thought, action, and sensing. So what happens is, to work for a neuron to work, they need oxygen to fire. So normally we call this neuronal firing, because actually when the scientists recorded the sequence of fire of neurons working, so it was like a firing sequence. So that's why they are called neuronal firing. So what happens is, when neurons need to fire, need to work, so they need oxygen, because oxygen is the source of energy for our in our body. So what happens is, hemoglobin is carried by oxygen in the blood vessels. So when I'm talking to you, it's not like a single neuron is activated. It's like a millions of neurons work together. So they fire together. So in different parts of my brain. So while I'm reading something, memorizing something, maybe my prefrontal area, the forehead, this area of the brain is being activated. The neurons, millions of neurons are firing inside. So what they need is, they need more energy. So when they need energy, what happens is they ask for oxygen. So the blood vessels, the blood flow goes to the brain and provides oxygen. So what happens is, the oxyhemoglobin is the chromophore that is used by neurons that extract oxygen from blood. And when that hemoglobin is given up to the neuron, it is transformed into deoxyhemoglobin. So in FNERS, we measure oxyhemoglobin and deoxyhemoglobin based on the near infrared spectrum of the light. So we will use two wavelengths of light, and then what we do is, based on the two wavelengths of light, we calculate oxyhemoglobin and deoxyhemoglobin. So here you see there are some sources and detectors. So sources are near infrared lights, and the detector can be any type of detector. Can be a photodiode, can be a phototransistor, or silicon photon counters. I will go in more details. So what happens? We place this montage over top of our brain. So what happens is, and then inside, as I was telling you, the blue ones are deoxygenated hemoglobin, and the red ones are oxygen carriers. So there is oxygen in there. So whenever uh, our brain is active, we need more oxygen. So there is an increase of oxygenated hemoglobin. So that's how actually the signals look like. The red one here is the oxygenated hemoglobin, and the blue one is the deoxygenated hemoglobin. So whenever you perform, if I perform some activity, some task, something to memorize or whatever, what happens is my blood oxygen will increase, and I can detect, okay, this part of the brain is active. So this is a simplified theory, a basic level that is used in this technology. So it is governed by the Beer-Lambert law, but this is actually used for light that is traveling through a medium in a straight line. So here you see I and I naught are the intensity of detected and incident light. So incident light is a light that can be shined through a laser light or a LED light, and a photodetector or any kind of light sensor detects lights. And mu is the absorption coefficient of the medium. But actually, in our scenarios, if this is a brain, so the light goes from the LED to a photodiode, and it goes all over the brain, but the photons that come back after making a journey, they go in a well-known banana shape. So we use modified Beer-Lambert law that takes into account the differential path and factor and unknown geometry. So because in the brain it is not as like a straight path. So we use these two factors to calculate the light detected. So that's how in the middle you see that's how light travels in the brain. So whenever you shoot light, it goes all over the brain, but it comes back. Some of the photons go deep, go there everywhere, and then some of the photons come back to the detector. So there are like multiple neuromonitoring technologies that you have heard about, like fMRI, EEG, but there are some advantages and disadvantages of fNERS. So fNERS offers higher temporal resolution than fMRI because it's fMRI, you are limited to a very slow response, and also fNERS has better spatial resolution than EEG. It is non-invasive, safe, inexpensive, portable, and simple to use. So these features are very helpful to use this technology. Especially this technology is much used in the infants and neonates. I will talk more about the implications. And then there are like three types of technologies: continuous wave technology, frequency domain technology, and time domain technology. So in my first part of the presentation, I will be talking about the device that are continuous wave. So what happens is they emit continuous intensity of light, and then measure light intensity at the detector. So that's how the intensity will for sure will decrease based upon the absorption and scattering. And then there is a frequency domain that modulates the emitted light intensity, and it measures the intensity as well as the phase shift in the signal. So if this is the light that signal that is going into the skull, and this is the signal that is coming out, so there might be a phase change, for sure there will be an intensity change. And then there's a time domain where extremely small short pulses of light of picosecond, and they shoot this light into the brain, and then they measure the time of arrival of photons. So what happens is you shoot photons, inject photons in the brain by like very tiny pulses of nanosecond, picosecond, and then on the other side of it's not, so these detectors are not simple detectors. You cannot use a small regular photodiode. So these are like single photon counter modules that are used. So here you see the spectrum where the HHB is the deoxygenated hemoglobin and O2HB is the oxygenated hemoglobin. So most of the near infrared spectroscopy devices, they works from between 650 to 850. So between this range, because at one side HPO will be higher, and the other side the deoxygenated absorption will be higher. So to measure both of these signals. So there are a lot of applications of FNS, like in behavioral and cognitive neurodevelopment, perception and cognition, hyperscanning, social interaction. It's especially used in psychiatric disorders, psychiatric conditions, and in infants, in speech and language studies, in rehabilitation and bedside monitoring. So you see, there are a lot different kinds of FNS devices that are currently used in different scenarios. Here you see uh, a device, a person performing a task. So what is he doing is he is performing some task. So while he performs a task, so we know, okay, at this time the task was shown, so that we know when we analyze the signals, that where actually the task started. So we know, okay, where we should look at the brain activation, where is the resting state. I will talk more about this paradigm. And there can be like multiple configuration that you can put your detectors and sources together. So, and then we are going to study functional connectivity. So functional connectivity refers to the statistical relationship between specific physiological signals in time. So if you consider this as a brain, each of the color marks a different region. So when we measure signals from different brain regions, they can be of any shape in any form. So the ideal signal that I showed to you, so this is kind of the ideal signal, but this is something that is rarely seen. So it requires a lot of pre-processing, filtering, and then we go to this signal. So what happens is we receive signals that are somewhere like this over a long period of time. So the idea is the synchronization between two brain structures, either at rest or during the performance of a specific task, reflects communication between these regions. So while I'm presenting to you, so it's not like one brain region is active. So there are many different regions in my brain that are at the same time communicating, and that's how I can use my tongue as a motor, because that my muscles in my tongue, they are activated. So everything that is working in my body is connected to my brain and has different location in the brain. So when we measure the signals from different locations, we find out, okay, if they have correlation between them. So how do we compute correlation is I will show you later also. So it is this, but so if two signals, let's say the green and the light brown color, they are like have a similar shape, so they are treated to have a higher connectivity. And if they are out, they don't have any similarities in the signal, so we say that this is a low connectivity between these regions. So the neuronal firing, and because of the neuronal firing, the changes in oxyhemoglobin and deoxyhemoglobin that we are detecting. So if these oxyhemoglobin signals or deoxyhemoglobin signals are matching to each other, we say it's high connectivity, and if they are not, it's low connectivity. And we are analyzing the functional connectivity together with the brain stimulation. So in this work, we are using transcranial direct current stimulation, the TDCS method. So here we have an anode and a cathode. So we all know that the current flows from the anode to the cathode, and because of that, if there are like two electrodes on, if you place two electrodes on top of your brain, so what happens is, if we the conventional stimulation that has bigger electrode size, and so they cannot confine. So you see, as a result, the electric field, the effect of electric field is all over the my forehead. So because of that, I cannot target a small portion of the brain. So in this kind of scenarios, we are giving stimulation to a large brain area. So So there can be multiple anode and cathode configurations, and because of the electrode size, the resulting electric field and the effect of brain stimulation can be different. So here you see there are 4 cathodes and 1 anode in the center. So what happens is the current goes from the anode to the cathode. So only a region, so we confine the electric field. So electric field is confined within this brain region. So only this area of the brain is stimulated. So now I will talk about the research that we did for to study the changes in brain connectivity. So we did this work while the person is resting. So this is also called resting state functional connectivity. So when I'm talking to you, my brain is activated. But if I am not talking to you, if I'm uh, doing nothing, so it is called I am in a resting state. So at that moment also I have brain signals. So at that time also there is blood supply to my brain. So there are fluctuations of oxyhemoglobin and deoxyhemoglobin. So there is still a there is still some communication going on in my brain. So based on that, we can still detect. So this connectivity type is also called the default networks, because this is something while I'm in resting state. So in resting state, we are supposed to do nothing. But in the resting state, if still there is communication, this means that this is like a default network in my brain. So the information, feedback, or anything, because we cannot shut down our senses. So my ears are working, my nose is working, and everything. I can control my motor, I can control my actions, but I cannot control the sensing, right? So because of that, my brain regions will be activated, and still there will be something going on. So we will study the effects of high definition anodal transcranial direct current stimulation on the prefrontal. So this is anodal. So when the anode is in the center, this is anodal TDCS. And we will use neuroimaging using fMRS on the prefrontal region. So we will analyze the resting state functional connectivity changes during the application of TDCS. So we are hoping to improve the executive cognitive function performance by increasing functional connectivity. Because by the literature, why we are doing this? Because from the literature, we know that, okay, the brain stimulation increases cognition. So in some of the researches, we already found, the researchers have shown that by providing brain stimulation, the cognitions or the dementia, especially in the old age citizens, it improves the brain health. And also it is used as a clinical intervention as a therapy in some of the clinics, if you check. And then the other thing is the functional connectivity. The connectivity, the higher the connectivity, the better the connectivity tells us the better is the cognitive capability of the person. Because if I tell you, like, Alzheimer's disease, is a disconnectivity syndrome. So in that disease, your brain is disconnected from, and they don't have a good communication. So you lose your memory, you lose the possibility to you perform daily tasks. So having this functional connectivity in our brain is good for our daily life. So this is the experiment paradigm that we will use. So all through the 25 minutes, the person will be resting in a position. So we have 3 phases of the experiment: pre-stimulation, and then we provide the stimulation, that is the HTTDCS, and post-stimulation. And we focus on the right prefrontal cortex, and we supply only a 1 milliampere of current, and we calibrate each of the cathode to receive an equal amount of applied current. So this is the channel configuration and instrumentation. So stimulation device, we use the StarStim TCS system with new electronic system. So we are using, out of all the electrodes, only 5 electrodes with 1 anode and 4 cathodes. So here you see the plus is the anode and minus is the cathode. So this is the forehead of a person, and on the right side you have this HTDCS, and the other numbers, the O and X, are the sources and detectors. So the device that we are using in this is Dynode by NIRX Medical Technologies. So in each of their optodes, they have a detector as well as a source, and they have combinations from each source to each detector. So we have total 14 optodes, and each optode plays both the role of emitter and a detector. So how we go forward to calculate the connectivity? So if these are the signals that we receive from our brain, so like I said in the beginning, this signal that I showed you is only for a short time and is an ideal signal. So actually something like this we will acquire from our brain. So what we do is we make pairs of all the channels. So each of this is coming from a different channel. So you see there are different channels in the 1, 2, 3. So a channel is between a source and a detector. So we have a total of 32 channels. So we have a total of 32 signals. So we will create a cross correlation matrix that will be 32 channels by 32 channels. So in this example that is from a paper is a 16 channels device were used. So here you see the intensity tells you that how good is the correlation between two channels. And then what we do is we binderize these correlation matrices. So from a correlation matrix, we go to the we using different thresholds, and these thresholds also depends on different literature and different priorities. So having a low threshold can give you a connection, can still give you some connection that are weak, and that can be just from some noise in the signal. So to have a good or to be confident that we can detect a good brain network, so we will use a higher threshold. In this research, we are using a threshold of 0.8. So whatever the correlation, if correlation is equal to or higher than 0.8, we will consider that as a connection. So one connection is between channel, let's say here, channel 7 and channel 5. So between two channels, we have a connection. So for this research, we are using graph theory analysis. So for graph theory analysis, we are using some commonly used metrics like connection density, nodal degree, nodal efficiency, global efficiency, global clustering coefficient. So these are some their basic mathematics, how they work. And then the connection density and the nodal degree defines actually the quantity of connectivity, because how dense is a network, or nodal degree tells you how many connections each channel has. So if each channel is connected to like suppose 10 channels, the nodal degree of that channel is 10. So these tell you about the number of connections. So this is the quantitative analysis. And then the global clustering coefficient and the efficiency, they actually describe the quality of connectivity, that how fast the communication is in the network. And perhaps these terms are much more, you have much more knowledge in the network and communication from me. And so here is the real signals. So as I showed you the signals, we have these channels from 3, 8, 9, 7, 1, 10, 13, 2, 14, and 12. So these channels are formed within the brain region where we try to stimulate the brain region. So here in the graph, you see the pre-stimulation place where you see there is no, there you see there is no, it's a baseline, and after the stimulation, after the 5 minutes, when the stimulation starts for 10 minutes, you see the right hemisphere, because right hemisphere was a hemisphere that was stimulated. So the stimulated hemisphere goes higher. The intensity of there's more oxygen in the brain. And then in the post-stimulation phase, wherever it reached, so if you see the trend, it increased, the HPO level increases, and then it goes straight. So these are the complete functional connectivity matrices. So here you have 4 quadrants of left hemisphere, right hemisphere, and then uh, this is on the left column is for the pre-stimulation, and then there's a stimulation phase, and then post-stimulation phase. So you see, if we binarize it, the red region, or if you binarize it, the white region has low connectivity during the resting state period of the pre-stimulation. And then when you stimulate the brain, there are different portions of brains that are activated. There are connections formed in, but at the end, when you go into the post-stimulation phase, the effect of stimulation has created more connections. So when you have stimulated the brain, there are many more connections that were, if you compare the pre-stimulation brain state with the post-stimulation brain state, you will see there are a lot of connections being formed. So if we analyze that, the increase that you see between pre-stimulation to post-stimulation in the right hemisphere, if we define a connectivity percentage out of the connected channels and based on the total number of channels. So in the beginning, there were only 11% connections, but after the stimulation, it has increased to 54%. And then in the left hemisphere, it also shows an increase. So from 23% to 61%. And then there are connectivity that goes from the right, between the right channels and the left channels. So of the right hemisphere of the channel in the right hemisphere to the channels in the left hemisphere. So you see everywhere there is an increase of the connectivity. In the right and left stimulation region, right hemisphere stimulated and unstimulated region also. And if you go to one-on-one, also there was no connection in the beginning, but at the end there was 60% connectivity. So connectivity in the right showed higher increase as compared. So although we have seen that, okay, everywhere there was an increase of connectivity, but in the right hemisphere, it was 4.5 times as compared to 2.5 times in the left hemisphere. And the interhemispheric connectivity was also increased 12.5 times. So highest increase observed in the interhemispheric connectivity was between the corresponding channels of the stimulated area. So the channels that were directly under the stimulation were the channels that were most affected. So this is what we were talking about, the effect of brain stimulation, that we have contained the effect of brain stimulation. So if you remember when I was showing you this part, so if we are focusing, if this is my stimulation, so the highest is also under the stimulated area, highest effect. And then if we plot the network matrix, so most of the graph theories like connection density, global degree, network clustering coefficient, and global network efficiency, all of them shows that the post-stimulation phase can be separated from the pre-stimulation. So everywhere we see a significant increase of connections from pre-stimulation to the post-stimulation phase. And in this case, only the clustering coefficient was insensitive to the phase information. So without, like, it doesn't matter of the pre-stimulation, so it was not much affected. But if you see the other one, they still do. So if you increase we have analyzed this through a range of thresholds. It's not like we do it only for 0.8 and if it is best, but we have seen that from 0 to 5 to 0.9, we have checked all the thresholds, but for the analysis, we only use 0.8 as our threshold. So you see the trends are similar for all the trends are similar for all the threshold levels from 0.5 to 0.9. And the individual node metrics, they also showed an increase, and then the highest increase is also under the channel that was simulated. So this, you see the nodal degree, so the number of connections that each channel has made from pre-stimulation, during stimulation, and the blue is the post-stimulation. So this is also this is also something that to highlight that nodal degree and the nodal network efficiency also showed the highest increase in the channels that were directly under the brain stimulation. And then we go to another. So after conducting this research, we moved to another one. So we saw that, okay, when we are analyzing this data, we hypothesized that, okay, the resting state function connectivity during stimulation does not increase after a certain duration. Because what happens is, when we are giving stimulation to the brain, although it is non-invasive, it is not painful, but still there is a tingling sensation on the brain and itching sensation on the brain. So while this is being used as a therapy for increasing the cognitive capacity of persons, or as a therapy, so we should know how much dose. So like if we have a headache, or if we go to a doctor, they give you a medicine of therapy. So but they also tell you that how much dosage you have to take. So in this scenario also, now we are trying to control the stimulation duration. So how long the stimulation duration should be, so that we can have the best effect. So because in the literature there have been stimulation duration from 3 minutes, 5 minutes, up to few minutes, like 20 minutes, 30 minutes of stimulation. So is this long durations of stimulation still necessary? Does this still affect the brain as they do in the beginning? Like if so, we hypothesize that, okay, after there might be a saturation point in our brain state. So when we are giving the brain stimulation to the brain, so there are connection forms, there are connection lost, and there are new connections made. So, but at some point of time, it will stay as static, or it is not changing significantly as it's changing in the beginning. So we designed a new study and a new feedback-based diagram and feedback-based strategy. In the previous one, it was an open loop that you give 5 minutes of rest and then 10 minutes of stimulation, and then 10 minutes of rest again post stimulation. We are using still the same data, but we are now analyzing what is happening during the stimulation phase. Previously, we say that, okay, before stimulation, the connectivity was low, and after stimulation, the connectivity was higher. That means our cognition has improved, or whatever we can infer from that. But do we really need that 10 minutes of stimulation, or is all of the patients, all of the persons are similar, are same, that they require same? No, because every brain is different. So there might be some people who reach some saturation point earlier, and some who don't. So we designed this. We provide a percentage of connectivity. We can provide a percentage of connectivity, or we can provide a brain. We provide a brain state that we want to achieve from the TDCS. So we have the TDCS controller that can control the milliamperes of current, and then we have a TDCS device. The plant is a human brain, and then we acquire signals from the FNOS technology. So these are the signals that can look like, and then we measure the brain connectivity. And over the period of time, we keep measuring the connectivity, and we make new matrices every instant and compare it with the healthy brain connectivity. That is this the connectivity that was already required by us. So if we have achieved that point, so we know, okay, we don't need further. So the hypothesis is the resting state functional brain connectivity during stimulation does not increase after a certain duration of time. The significance, it is very significant because effective duration of stimulation can be provided in neuromodulating the diseased brain. So if only effective duration, so we don't need the extra minutes of stimulation, so we can avoid longer therapies, we can save time, and also there is some suffering caused by the patient or the subject, we can also remove that. So if we analyze the percentage connectivity during stimulation, the channels that are considered connected only when the value of PSM connection is greater than 0.8, we consider 0.8. And then we computed the percentage of connectivity. You see here, green is the complete prefrontal region. Red is the right hemisphere. Remember, the right hemisphere is this hemisphere that we are actually stimulating. And then the left hemisphere is cyan, and magenta is the right to left hemisphere connectivity. So you see, this is the 10 minutes of only the stimulation. So you see it from time from 0 to 600 seconds. And we treat 3 minutes as a stabilization period that, okay, we can fix, okay, at least 3 minutes everybody has to go through this stimulation so that we have a standardized protocol, we can have a standardized analysis. So we will have a stabilization period of 3 minutes. And after that, you see the red graph goes much higher in the beginning, and then it's not increasing a lot. Although they are remaining, they are still growing, but the right hemisphere, that is the hemisphere that is under the stimulation, is not significantly increasing. So if our, so in this case, what happens is if our target, if our target was only we want, because we are using, remember, we are using the HD high definition TDCS. So we wanted to contain the effect of stimulation to a brain. So we know, okay, this is the brain region of the person that needs rehabilitation. So we only need to provide stimulation to this area. So in this case, the right hemisphere is the region that is our region of interest. So we see that, okay, the right hemisphere has not increased. So you see, after like 6 minutes, the last 4 minutes is not increasing a lot. So this means that, okay, if in case right hemisphere was the target, so having 10 minutes of stimulation was not a good idea. Only 6 minutes of stimulation was the 6 minutes was the effective duration. So the increase in connectivity during electric stimulation was highest in the stimulated hemisphere. That also this we showed previously in the pre and post analysis, and also during the stimulation also. So if you see the nodal degree, if you see the it goes higher and then it's not increasing a lot. So these are the short range channels of right hemisphere, short range channels of left hemisphere, and then the long range. So in networks or in connectivity, there are like some nodes who are already close together. They have a, we are treating, so we have two hemisphere, right hemisphere and left hemisphere. So inside right hemisphere, we call the connections made within the right hemisphere the short connections inside right hemisphere, and the short connection inside the left hemisphere. But still there is a possibility, because as I said in the beginning, the brain is a complex organism. So there can be communication between different parts of the brain, because we have never gone through, or we have never understood brain until now, till date, completely. So we are still learning. So there are still points they can, and we find out that in this research as well, that there are some regions in right hemisphere and left hemisphere that are connected. So these are the graphically shown images that shows the density increasing over the time period of stimulation. So you see it increases its complete prefrontal connectivity. So from 17%, it goes to 18% and 25 but it keeps going increasing. So if we analyze whole prefrontal connectivity, all the channels that we measure, so we see that it keeps on increasing. And then if we treat only the long range connectivity, that is from one hemisphere to the other hemisphere, we see that okay, it's also increasing. The connections are also increasing. And then if we treat the short range connectivity, you don't see the connections from one hemisphere to the other. So now we're talking about short range connectivity. So if you see the right hemisphere, it goes from 32% to 50% in 6 minutes. And then for the last 4 minutes, it only increases to 5 more percentage. But in the left hemisphere, you see there is a lower increase of connectivity, but still there is a gradual. But our target was the right hemisphere. So okay, by this research, we are also I'm running short of time, so I will try to be. Then from this research, we also made an inference. Okay, it's now clear that okay, by 6 minutes, there was an 18% from 32 to 50%. It was already achieved in first 6 minutes, and only 6% was increased in the remaining 4 minutes in the right hemisphere. So we have seen that high density stimulation has focal effects on hemodynamic response as well as functional connectivity in prefrontal cortex. And then the new brain connections are made faster in the targeted brain region during HTTDCS. And brain state does not show significant improvement after reaching a certain level. So this can be because in this case, our region of interest was the right hemisphere. And then neurofeedback-based real-time functional connectivity can be used to control stimulation duration and noise and overdosage. So this algorithm, this analysis that I have developed, so it is kind of real time. So FNS signals are also measured in real time, and measuring this Pearson coefficient is not a cumbersome task. So it can also be computed in real time. So in real time, we can analyze and stop this functional activation. So now I will briefly go, because I was telling you about the NUS devices as well. So this is what we have done with NUS devices, but I have been developing the NUS devices along with this brain research with stimulation. So this is the wirelessly operated device. This is a block diagram. I will not go into details, so I will just, so it has a simple controller, it has Wi-Fi, and then it has a possibility that it can include multiple electrodes from EEG as well. Then there can, as I showed you the spectrum, we have used the different light sources. I will go. So this is the high density configuration of NURS. So here you see we have this box at the bottom. You can see each square is 5 millimeter cross 5 millimeters. So the idea is this modular NURS device can be, we can have multiple modules and we can cover major brain regions. So, and what we do is when we measure through different source detect separations, so we have separation of 5 millimeter, 20 millimeter, 25 millimeter, 30 millimeter, and 35 millimeter. So we have these radiuses. So there are some channels that are between these lengths. So when if you remember in the beginning that the banana I showed, so the depth of the banana depends also on the source detector separation. So higher the source detector separation, the deep we will go in the brain. So as you see, we have different depth information in each of the circle. So we can make a 3D image out of this. Like this, simulated. So we have in short separation channels. Short separation channels are very important in the brain study because we still have the above our real brain, there is multiple layers of skull, superficial layer, and then there is, so there are multiple layers of in my head. So before I reach actually the brain. So the long channels they go, but the short channel they don't go deep. So only they carry information of my skull and my skin and skull. Um, I will just this is a video of like how actually this is a slow motion video of how it is. It's very slow, but because actually it is this whole frame, whole round is completed in at 50 hertz. So this is very slow. So this is how it works. So we shoot one wavelength of one LED at a time. So it is time multiplexed in the switching of LEDs. And then in this, because human brain and body is different, so every it can take more light. It might need more light for my head as compared to somebody other head. So we have to calibrate the intensity of light as well. So we have I have designed this as well. So the intensity of light can also be calibrated based based on the requirement. So this is actually how it looks like. So it can go in a backpack. This is the device, which is embodied in the 3D printed material. So inside the case, and then this is a fishing cap that sorry, swimming not fishing cap. It's the swimming cap that has this device embedded inside. It is a flexible so that it can go to on our head and it can block the light as well. I will just so these are the some tests to verify the working of the device. So the regular tests that are commonly used are the occlusion test, because in occlusion we stop the blood, so it is very simple, so that we don't have more blood supply, so we can detect the oxygenation and deoxygenation very clearly. So we did this test, and it's a hand tapping test. Test is also like it's a very generic and very basic test in FNRS that we do. It is a standardized, and you see a response. And also based on the short separation, we can improve our signals. So the enhanced is a dot one. So if you see, if you remember the initial signal, the dotted signal look much like the initial signal. There are some phantom tests. Phantoms are the solid objects that are solid objects that can be used, that we know the absorption and the scattering coefficient. The material, we already know what material is inside that phantom, and we measure uh, through those phantoms to verify the device working. So all the tests we did showed good response. Developed a software that can provide you 3D map, also the signals. So and this work until here, the development of this this was the continuous wave device and the brain connectivity. This is the work that I did in Busan National University, Korea. And now during the last years, I have been working on a project called VASCOVID. It is a hybrid diffuse optical device for the assessment of microvascular health at the intensive care. So we worked during the COVID with in intensive care of hospital. And this is a European project that has multiple partners from all around the Europe. So in this device, I will show you. So we have international consortium, we have academic and research institution, biophotonic companies, regulatory companies, and clinical end users. So these are the modules. So in this case, we are, if you remember in the beginning, I showed you the nurse types in the there was a time domain nurse that has small pulses of light, and we detect the photon counts. And then also we have diffuse quotient spectroscopy, and we have automated tourniquet, and we also measure the heart rate through the pulse oximeter. So this is how actually the device looks like. And I will just skip everything and show you a video that So this is a device working in the hospital. It's a, if you see, so it's a ICU room, and where we are measuring a patient. He's on a ventilator. So the idea was that we are using this device to measure COVID patients, that were how good their blood circulation, how good is their oxygenation, oxygen concentration in the blood. So we place these probes. We wrap it on the muscle in the forearm, plus oximeter on the other arm. So this is the software device and a embedded software that is inside. So that tells the doctor who is working on the patients what are the how the device is working, and how is the vitals of the person, how the signals, the blood oxygenation parameters, blood flow index, hemodynamic parameters. So this is the technology that this is very accurate technology. The continuous wave that I have developed previously and worked with, it lacks in the precision and accuracy. It can only measure oxygenation, and that is also changes in oxygenation mostly. They claim to use the special resource spectroscopy, and then they can measure absolute values of oxyhemoglobin and deoxyhemoglobin. But with this Vascovid device, we can measure accurate measurements, absolute measurements of blood flow, of the hemodynamic, because with this technology, we can actually measure the how much is the absorption, how much is the scattering. We can measure everything, and then by combining all of these, we measure metabolism, that how much blood flow is going and how much blood flow is being consumed. So this device, as you see, is not being used on the brain. So it is any tissue. It can be brain, it can be any tissue. So it detects the blood flow, it detects the oxygen saturation, and based on that, we can measure the metabolism. So it has like wide applications in intensive care units that we are using. We are already working on multiple different protocols. By the time we developed this device and we talked to this intensivist who was working with us, and then we already are coming up with new studies. So although the project, the data collection is finished, but we are still measuring different patients in different protocols now. We have used similar devices in neurocritical care, neonatology, vascular diseases, and also in sports. All right. Thank you.